Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing DBT BET JRF 2024 entrance exam question paper. If you are new to my channel, please do like, share and subscribe. I have also uploaded still previous year's question paper of DBT BET JRF. You can go to my channel playlist and there you will find a folder with DBT BET JRF. In that you will find the question papers which I have answered. Then next one. Real-time PCR was done with two patient samples A and B to detect SARS-CoV-2 virus. The CT value obtained in a patient sample A was higher than that obtained in patient sample B. The inference to be drawn from this observation is that which is having higher viral load you have to see. So CT values, they are inversely proportional to the amount of the target DNA or RNA sample, RNA in your DNA or RNA in your sample. They are inversely proportional. That means lower CT value indicate high amounts of the nucleic acid, and higher CT value means lower amount of nucleic acid. So here the CT value obtained in patient sample A was higher. So higher CT value means lower amounts of nucleic acid, and lower CT value, that is sample B is having lower CT value. So that patient B is having higher viral load than patient A. Answer is option 2. RNAi was discovered in Caenorhabditis elegans. Answer is option 2. Which of the following statements about neutrophils is incorrect? They are the most abundant circulating leukocytes. Yes. They differentiate in the bone marrow and move into circulation. Yes. They differentiate only during bacterial infection. No, they are part of the innate immune system. So they differentiate during inflammation, then bacterial infection, then environmental exposure, as well as in some cancers. So 3 is the incorrect statement. Fourth one, if you see, they are recruited to the site of infection in response to chemokines. That is also correct. The answer here is option 3. Then next one. Generation of a DNA probe using random primer technique uses a combination of oligonucleotides 6 base pair in length. So you have to generate a DNA probe by random primer technique, and the oligonucleotide, that is length of the probe, is 6 base pair. How many distinct oligonucleotides are possible if all 4 nucleotides are randomly incorporated? Now all the 4 nucleotides, that is A, T, G and C, could be incorporated randomly. So there are 4 nucleotides and oligonucleotide of 6 base pair in length. So you have to calculate 4 raised to 6, that will give you 4096 as the answer. This means that 4096 possible distinct oligonucleotides, each of 6 base pair long and with all 4 nucleotides randomly incorporated, can be formed. Now how did you get 4 raised to 6? So if you have a sequence like A, T, G, C, D, G, T, now what is the probability that the first position will be occupied by a nucleotide? So there are 4 possibilities. Now what is the probability that the second position will be occupied by T nucleotide? There are again 4 possibilities. Then third position occupied by G will be again 4. Fourth position occupied by C will be again 4. Like that, if you see, there will be 4 raised to 6 possibilities. 4 into 4 into 4 into 4 into 4 into 4. So totally 6 possibilities will be there. So 4 raised to 6 will be 4096. Then next one. Which one of the following is likely to happen when a double-stranded DNA solution is heated? So when it is double-stranded DNA solution is heated, it becomes single-stranded, so absorbance increases. So the absorbance of DNA at 260 nanometers increases. DNA absorbance of DNA is measured at 260 nanometers. Then, which one of the following is not primarily a microtubule based structure? It is philopodia. It is an actin filament. So answer is option 4. They have asked not a microtubule based structure. Then next one. There are 20 amino acids. How many different polypeptide chains of n number of amino acids are possible? It is given by the formula 20 raised to n. So the answer here is option 4. Which one of the following organism require a biosafety level 3 facility for culture and manipulation as per extant biosafety regulation? So as per biosafety level 3, laboratories, the laboratories which have biosafety level 3 facility are used to study infectious agents or toxins that may be transmitted through the air and cause potentially lethal infection. So out of these options, Streptococcus pneumoniae is an airborne one. So it requires biosafety level 3 facility for culture and manipulation. So the answer here is option 2. Then next one. The process by which the genetic material can be transferred from one bacterium to another by a virus is known as transduction. Answer is option 3. Next question. In a newly discovered type of genetic material with similar properties to our own DNA, there are 4 nucleotides LMNO. L and M are complementary and paired with one hydrogen bond between them. N and O are complementary and paired with 2 hydrogen bonds between them. Given this, which of the following sequences would have the highest melting temperature in their double-stranded form? So highest melting temperature in our normal DNA is for GC bonds. Why? Because GC bonds have triple hydrogen bonds between them and it requires higher temperature to break the GC bond. Now here N and O are having 2 hydrogen bonds whereas L and M are having only 1 hydrogen bond. So it will be difficult to break the N double bond O and it requires higher temperature to break N and O double bonds than L and M double bonds. So in the options you have to see for which DNA is having the maximum number of NO bonds. So first one, if you see, it is more of L and M. Second one is also L and M. Third one is having O and N but it is lesser than that of fourth one. So the answer here will be option 4. Then next one. DNA polymerase 1 from Escherichia coli lack which one of the following enzyme activities? So it lacks the 5' to 3' RNA dependent DNA polymerase activity. Transmission of organism from mother to fetus or newborn child is known as vertical transmission of infection. Which one of the following is the most likely to transmit vertically? It is Chlamydia trachomatis. Answer is option 2. Then next one. Which one of the following combination of drugs act to inhibit the same metabolic pathway? So the answer here is sulfonamide and trimethoprim. They both are blocking the synthesis of folate. Now how does sulfonamide do is it inhibits the enzyme dihydropterate synthase. And trimethoprim, it is a direct competitor of the enzyme dihydrofolate reductase. And both these inhibited enzymes, they are involved in the synthesis of folate. So sulfonamide and trimethoprim, they inhibit folate synthesis, and that is, as per the question, they inhibit the same metabolic pathway. So the answer here is option 1. Now, which of the following statement is not correct about human immunodeficiency virus? HIV is an enveloped RNA virus. That is correct. Acyclovir inhibits HIV replication. No. Acyclovir has been shown to inhibit HIV replication under in vitro conditions, but not in vivo because of certain factors like low potency, high toxicity, and development of resistance. As a result, acyclovir is not recommended for treatment of HIV infections. Instead, a combination of antiretroviral therapy is the standard care for HIV treatment, and it includes a combination of drugs from different classes such as nucleoside-nucleotide reverse transcriptase inhibitors, non-nucleoside reverse transcriptase inhibitors, protease inhibitors, and integrase inhibitors. So acyclovir is not inhibit HIV replication. So the answer here is, they have asked which is not correct. So answer is option 2. Then next one. In India, major burden of tuberculosis, which is caused by Mycobacterium tuberculosis. Efforts are on to eliminate TB by 2025, and effective prophylaxis against the disease can be achieved with the BCG vaccine, which has been developed from. So BCG vaccine, it is developed from Bacterium bovis. It is an attenuated live bovine tuberculosis bacillus that is used in the production of vaccine. Then next one. Flow cytometry is an analytical technique that quantifies the frequencies of cell binding to fluorescent antibodies and scattering light in characteristic ways. When a flow cytometer is used to sort cell subpopulations on the basis of fluorescence and light scattering, it is referred to as fluorescence activated cell sorting. Which one of the following statement is not correct regarding fluorescence activated cell sorting? First one. Every time a cell passes in front of the laser beam, light is scattered and this scattering of the laser signal is recorded. That is correct. The more forward light scatter. Now before that, there are 3 types of scattering, that is side scatter, forward scatter, and backward scatter. So forward scatter is what they have given in option 2. The larger the cell, and so the amount of light scattered in the forward direction can be used as a rough measure of the range of sizes of the cells in the stream. That is forward scatter, what they have given in option 2 itself. What is side scatter? It measures the light scattered at a 90-degree angle, which is related to the cell's internal complexity and granularity. And what is backward scatter? It measures the light scattered in the backward direction, which is also related to the cell size and shape. So option 1 and 2 are correct. Now coming to third one. The amount of side scattered light offers an indication of the extent of size of the scattering cell. No, they give an idea about the internal complexity and granularity. So 3 is not the correct statement, and your answer is option 3. Now fourth one, if you see, cells in suspension are hydrodynamically focused into a narrow stream by being introduced inside a rapidly moving column of sheath fluid. Yes, that is correct. And that process, it is called as hydrodynamic focusing. And this technique, it enables the cells to be precisely aligned and spaced, allowing for accurate analysis and sorting at high speed. Now coming to next one. While running in a 200-meter race in national games, the required ATP generation in the athlete is primarily facilitated by. Now there are two types of ATP generation in such conditions. One is anaerobic glycolysis. Glucose is converted to pyruvate and ATP generating lactic acid. Anaerobic glycolysis is fastest way to generate ATP but is less efficient because it produces lactic acid which can result in fatigue. So they have not given regarding anaerobic glycolysis in the option. So the second one by which ATP is generated is phosphocreatinine breakdown. It is a high energy compound stored in the muscles, and during intense short duration activity, phosphocreatinine is rapidly broken down to generate ATP. So the answer here is option 3, that is creatine phosphate in muscles. Then next one. A slide of macrophages was stained by immunofluorescence using a monoclonal antibody for the TAP1-TAP2 complex. Which of the following cellular compartments would exhibit positive staining with this antibody? So TAP1 and TAP2 complex, it is involved in antigen processing and presentation, and it is located in the endoplasmic reticulum. So the answer here is option 2, that is endoplasmic reticulum will give a positive stain with this antibody. Now next one. Agglutination is a simple, inexpensive, rapid, highly specific immunological test that is widely performed in diagnostic laboratories. For example, it is often used for human blood typing based on the presence of specific antigens on the surface of red blood cells which vary among individuals. That is ABO, human blood typing. These are the various blood which humans have. Blood groups. Then agglutination is routinely used in clinical laboratories for determining HIV infected CD4 plus cells. No. For detecting HIV infected CD4 plus cells, you use quantitative real-time PCR, not agglutination. So statement 1 is correct, but statement 2 is incorrect, and your answer is option 3. Then next one. The vaccine for cervical cancer is composed of. So it is composed of HPV, human papilloma virus-like particle, VLPS. VLPS is virus-like particles. So the 9-valent HPV vaccine, it is a non-infectious recombinant vaccine prepared from purified virus-like particles, that is VLPs, of the major capsid L1 protein of HPV. Now next one. Cystic fibrosis. Cystic fibrosis is for CFTR. A2. Then Leishman syndrome. It is hypoxanthine guanine phosphoribosyl transferase. B1. Severe combined immunodeficiency, adenosine deaminase. C4. Spinal muscle atrophy is SMN1 or 2. Answer is option 4, A2, B1, C4, D3. Then next one. Which one of the following antibiotics specifically inhibit RNA synthesis? First one, isoniazid. It inhibits mycolic acid biosynthesis, and this leads to inhibition of cell wall formation and ultimately death of the bacterial cell. Now penicillin, it is also an inhibitor of bacterial cell wall synthesis. Streptomycin, it binds to the 16S rRNA located on the smaller 30th component of the bacterial ribosome and inhibits peptide bond formation. Rifampicin, it inhibits the synthesis of RNA and because it binds to RNA polymerase. So they have asked for the antibiotic which specifically inhibits RNA synthesis, so it is rifampicin. Answer is option 3. Then next one. If one parent carries the defective Huntington disease gene, his or her offspring have 100% chance of inheriting the disease. Huntington disease is autosomal dominant genetic disorder. Yes, that is correct. But statement 2 is not correct because only one of the parent is carrying the disease, the other is not carrying. So only 50% chance of inheriting the disease is there. So statement 1 is incorrect but statement 2 is correct. Answer is option 4. Then next one. Functional magnetic resonance imaging (fMRI) is one of the most powerful methods for examining brain function. This method is based on the changes in the magnetic properties of. So fMR1, it is based on the changes in the magnetic properties of oxygenated and deoxygenated hemoglobin in the blood. So the answer here is option 4, hemoglobin. Which of the following diseases is due to severe deficiency of proteins in the diet? It is kwashiorkor. Scurvy is vitamin C deficiency. Tay-Sachs disease, it is due to mutation of the hexa gene or the hexosaminidase A gene. Then myasthenia gravis, it is an autoimmune disorder and it is caused by the production of self-antibodies that block the receptors of acetylcholine. Then next one. Given below are two statements. One is assertion, other is reason. Assertion. Mycoplasma stains negative in Gram staining. Yes, that's correct. Mycoplasma cell wall is devoid of teichoic acid. Mycoplasma doesn't have a cell wall. They lack cell wall containing peptidoglycan and all. So A is correct but R is not correct. And the answer is this is more opt answer. Answer is option 2. Both A and R are correct. Yes, R is also correct because they are devoid of teichoic acid. Teichoic acid is a component of the cell wall. So A and R are correct but R is not the correct explanation of A. Then next one. Which one of the following is responsible for the development of myelin sheath in central nervous system? It is oligodendrocytes. Answer is option 2. Which one of the following brain regions are not involved in eye movement control? So it is inferior temporal gyrus. Answer is option 3. Which one of the following disorders lead to hallucination? So it is schizophrenia. In absolute refractory period of neurons. Absolute refractory period is the time in which neurons cannot fire another action potential. So Na plus channels, they are open. Answer is option 1. Phagocytosis in the central nervous system involves. So usually phagocytosis in central nervous system, it plays role in maintenance of tissue homeostasis, defense against infection, clearance of apoptotic cells and debris, modulation of inflammation, regulation of the immune response. Sorry. So out of these, only clearing of dead cells is correct. Others like bridging the blood-brain barrier, specialized ability of some neurons to divide, recovering excess A2 plus from synapse, all that is not part of the phagocytosis in central nervous system. So answer is option 4. What attribute do DREB transcription factors impart to higher plants? So DREB transcription factors, they play a crucial role in plant response to drought, heat and cold stress. So the answer here is option 3, drought resistance. Then next one. Quantitative trait loci can be mapped using single sequence repeats markers, SSR markers. They are popularly used in QTL mapping because of their high polymorphism, easy amplification and codominant inheritance. Then next one. Heterologous expression of prokaryotic genes in plants can be used to render resistance against insects. That is correct. And what is heterologous expression? Heterologous expression, a gene from one species is expressed in another species using genetic engineering techniques. Then statement 2. Expression of Bt cry1ac gene in cotton improves resistance against aphids. It is not aphids, but it is against the lepidopteran pest cotton bollworm. It doesn't provide resistance against aphids. So statement 2 is incorrect, whereas statement 1 is correct. Statement 2 is incorrect. Statement 1 is correct. That is option 3.